also particularly happy to be presenting Dr. Ewan Carr. He's going to be talking for us under the title of Identifying Homogeneous Subgroups of Patients and Interesting Features, a Topological Machine Learning Approach. And as I said, I'm particularly happy to be presenting him because I've been working with him, and it has been a very big pleasure to work with him along the, I would say, almost 3 years, Ewan, probably. Or even more, and in an area on topological data analysis where I really do base most of my research and that I'm very interested in. And Dr. Ewan Carr is a BRC research fellow in our department here at the Biostatistics and Health Informatics Department at King College London. He completed his PhD in social statistics at University of Manchester, and he has been previously based in epidemiology and public health at UCL. And he has a broad research interest, so you can see him working on latent variable models, on predictive models, and recently he has been also developing studies on COVID research. So he's a very a person that can work with very different techniques in statistics, and also with topology and topological data analysis. That will be the work that he will be presenting today. And so, Ewan, when you want, so I leave it to you. And after the talk, all the audience will have the chance to make the questions that you may have. Thanks very much.
Thank you, Raquel. Thanks for the introduction, and thanks everyone for coming and spending an hour on your Wednesday afternoons. I think Raquel's given a good introduction to the sort of this project. This is one of a number of projects using topological data analysis that we've been working on in the last couple of years. So I'm going to start with a sort of 30-second version and then a 2-minute version, because in the middle of this talk, I'm sure I'll lose some of you, but hopefully we can come back at the end. So in brief, topological data analysis allows us to study the shape of data. And we are using this, sort of building on some existing software. We wrote a pipeline in Python to identify clusters of patients. So that's what I'll be talking about today, and that's what we're doing. The sort of slightly longer version of that is that in recent years, I guess we're talking the last 10 or so years, there's been this growing interest in topological data analysis, and this set of tools, we're just going to use some of them today, but the set of tools to infer and sort of really use the shape of data. One particular algorithm I'll be talking a lot about today is called Mapper, and this takes a high-dimensional data set and reduces it down to a one-dimensional graph. And from this graph, we can identify these representative topological features. I'll talk a lot about that later on, and we can look at sort of clusters within these features. This approach in general, so TDA has a number of advantages, and I think also our pipeline sort of has a number of features that I think are valuable. There are also some challenges that I'll get to as well. And this is all written in Python. It's on GitHub, and we're building on some existing open source software. Okay. So I should say I'm going to talk a bit about TDA and Mapper. I'm then going to talk about a couple of extensions that we are sort of making to Mapper, and then I'll go through the pipeline itself, and then we'll conclude with sort of some lessons learned. So this work is based on this paper that came out recently in BMC Bioinformatics. This paper describes the pipeline, and there's sort of a accompanying page on GitHub with the code, and that's essentially what we're talking today. So both of these are available. This is work done as part of the working group on topological data analysis for machine learning. So alongside with Raquel, there are a few others of us here at King's, but also we have external collaborators. And I've been involved with a couple of projects, or a few projects related to TDA. The one I'm talking about today, all about clustering and Mapper, but we're also elsewhere looking at incorporating TDA into prediction models and how we can use TDA to incorporate repeated measures data into prediction. So that's for another day. I have this slide here because when I started looking at topological data analysis, I knew nothing about topology. I'd like to say that's changed slightly, but I'm definitely not the expert. I've been fortunate to have Raquel sort of leading this project, but also to have some expert collaborators who do know topology and actually have been invaluable in sort of this process of me trying to absorb as much as possible. But I will do my best. Any mistakes are definitely mine. Okay, so talk about a little bit about topological data analysis. Now, you may have seen in January, Raquel gave a talk that gave a very nice introduction to topological data analysis or augmenting machine learning for TDA. It's on GitHub. So I won't be repeating that, but I will try and sort of point at some of the key developments. So this is a growing field. This is just showing the number of papers on topological data analysis in the last 20 years. And I think that the growth that we're seeing more recently is more sort of applying this to, well, in our case, to biomedical research. How can we use topological data analysis in machine learning or prediction or whatever else it is? So trying to translate these ideas out of topological mathematics and into more applied work. At least that's certainly my interest. Some key developments in this period include the idea of persistence and persistence homology. So we won't be talking much about that today, but sort of landscape diagrams and persistence landscapes. The map algorithm, I'll be talking about a lot. And then in recent years, sort of incorporating topology into statistical analyses and using things like a bootstrap, which we'll be talking about, or incorporating this into a sort of machine learning workflows, particularly in the last couple of years. So huge literature, really sort of exciting and broad applications and potential here. We're just going to focus on MAPA today. Okay, so the idea that we are starting with is that a data set that you have has some shape, shape that is invariant under small transformations. I'll come back to that in a second, but I found this quote online that is, I think it was taken from a blog post, but it made sense to me, so I'm going to sort of read through this now. So topology is not concerned with the exact geometric representation of the object, and it's not really bothered how many edges an object has or whether it's round or oval in shape, but rather it's going to try and describe the object in more general terms. So how many distinct components does it have? How many holes or how many cavities? So we're looking for something about the shape of data that is sort of invariant and that we can sort of change, well, some of these aspects of the data of the shape, and we're still left with the same topological structure. So some of these concepts that Raquel talked about in January include coordinate invariance. So the properties do not change with different coordinate systems. Deformation invariance, so we can stretch or squash the shape while maintaining its features. And then the idea of a compressed representation. So we should be able to simplify a shape down to just its essential features, but retain that shape and retain the components of that shape. So my brain works more in pictures, as you'll see throughout this talk. The example that we often see is this coffee cup and this donut. And to me, these are very different objects, but in a topological sense, we might just identify this loop or this hole. And in that sense, these are both have the same topological structure. In case you don't believe me, thankfully somebody online with some artistic ability has drawn this sort of, or has modeled this, showing that we can sort of reshape or sort of deform, stretch this donut and back into the cup. And throughout all of these stages of this transformation, it still just has one hole. And so from a topological sense, it is unchanged. So this is from, well, we'll come to MAPA in a second, but this image here is showing sort of alternate representations. So we have a horse in two different poses. We have a human head with different expressions, although that's quite hard to see. And then we have an elephant. And then I think we have some sort of flying elephant. And the point here is that although the sort of structure, what it appears as though the structure has changed, you know, the pose of the horse is different, or the facial expressions are different, the underlying topological sort of representation of that, shown on the right-hand side of each of these, is unchanged. And so we can sort of we can make this elephant fly, but the topological structure is unchanged. And so this is an idea we'll keep coming back to, because this is going to be really useful in when we're looking for clusters in our data set, and looking, for example, in antidepressant treatment response. We're looking for clusters that are defined by invariant topological structures. I saw this in another talk, and I thought it was too good not to borrow the references down the bottom. This is Picasso in 1945. I don't think he was talking about topology at the time, but this is a very nice sort of representation of what we're getting at. So in the top left, we have 9 pictures of a bull, and in the top left, we start with the most detailed picture, who's definitely a bull. And as we move down this diagram, the representation becomes more and more sort of simplified and just reduced down to the sort of the essential elements of a bull. So it's still recognizable, but we have sort of stripped away all the bits we didn't need, and we have come up with this simplified representation. So this is what we are trying to do with MAPA and with TDA. We have a huge data set that is high dimensional. We want to extract just the essence of the shape of that data. So you might be wondering, you got to this point in the talk and you hear about sort of, I don't know, clustering in biomedical data sets. You might be wondering, what's with all the elephants? What with the 3D shapes? How is this actually connected to something we might use in practice? I agree with you, and that was my thinking, sort of trying to absorb this literature early on. It was sort of, okay, I see the shapes and elephants, but what does that mean for our data? So hopefully the rest of this talk will come to this. So a couple of examples of how this has been used. So this is one looking at different, identifying different subgroups of breast cancer. And so this is a graph that has been generated from a large high-dimensional data set that allows the researchers to identify these particular types of breast cancer with particular properties. Another one slightly more recently, looking at type 2 diabetes, and so it's identifying these sort of hotspots, these clusters, that based on this graph that draws on this large high-dimensional data set. So, so our starting point then is we were interested in sort of heterogeneity in treatment response. So I'm not really going to talk about data, sort of specific data sets today. We were using GenDep, so we have a large amount of genetic, clinical, and demographic data from participants over a 12-week trial of 2 antidepressants. So that was the context, and that was what we sort of have initially applied this to. And we want to use topological data analysis to identify clinically relevant subgroups with differing prognostic outcomes or clinical profiles. And today, the focus is very much going to be just on this pipeline. So there are no results or very few results. It's really about how does this work? How does this all come together? And then maybe another time we'll be talking more about the sort of applied paper. So this paper was just about the pipeline and just sort of producing the code. Okay, so at the same time, we wanted to make some improvements. And some of these improvements have come from by building on MAPA and building on existing software. And some of these are sort of more about the way that we've gone about this. And so parameter selection via grid search rather than just sort of trial and error. Focusing on topological features rather than just nodes. So one of these previous examples, we essentially are coloring in just these points based on some variable, and then just circling the relevant these clusters. So we wanted to really return to those sort of invariant features, the hole in the cup and the donut. We want to find that in our data. And then lastly, we wanted to use statistical summaries. So we're going to use XGBoost and other statistical summaries to interpret the features and the clusters and the sort of membership to these features. And we'll come to that later. So in the next sort of 20 minutes or so, 25 minutes, I'm going to introduce the mapper algorithm. I'm going to go through a couple of the extensions or things that we have added on, and then I'll sort of go through the pipeline. So lots of setup, and then the actual bit about the pipeline will be quite brief, I think. Okay. So mapper is an algorithm. It's associated with this paper you can see on screen now from 2007 by Gunnar Carlson and colleagues. So that was sort of the starting point. And mapper is an algorithm that takes sort of a complex data set and reduces it down to a one-dimensional graph. So the starting point is a finite point cloud for which we can calculate distances between any 2 points in that point cloud. So let's have a real example. I'm still with the 3D models, so I'm still going to be explaining things in terms of 3D models because that's what most of the literature is based on. But I will try and relate this back to our sort of more applied clinical example later on. So suppose we have a point cloud. We have a cloud of points for a human hand. The first step in mapper is then to color in the points based on a filter function. So this filter function I'm going to say quite a lot about because it's really important and it's really, I think, powerful in how we use mapper. So the filter function is essentially just a function that maps any point in your point cloud to a real number as we see here. So for example, this is, again, simplified, but imagine each of these points that we had previously has an X, Y, and Z coordinate. And our filter function that we're going to use here is simply just to take the X coordinate of each point. So we would map each point that we saw earlier onto this line along the X-axis. So once we've applied our filter, and I'll talk more about the filter later, we then are going to split this into bins. So we've taken that sort of length of the along the filter, and we've divided the points in our point cloud into these bins with several intervals. We then carry out clustering within each bin. So this is somewhat counterintuitive because we started out looking for a clustering algorithm. We're actually now going to use a clustering algorithm like dbScan or another clustering algorithm of your choice within each of those bins. So just to recap, you start with data points, you define some filter function, you divide along the filter function, and now we're going to sort of carry out clustering within each of those bins. We then connect having carried out clustering, we then connect nodes that have the overlap in terms of their data points. So in the case of this 3D model of a hand, it would be if some of the points for this finger are also found in the node for this central bin here, and so on. And then we sort of remove everything else, and then we are left with just that network. So that's mapper. You start with a point cloud, some data. You choose a filter. You divide that filter up into intervals and have these sort of bins of data. You carry out some clustering, and then you connect nodes where they overlap. And you are left with this simplified representation of that set of data points. So at this point, you might think, great, we've ran mapper, and and we've got a nice colorful picture. And we could just stop there. So each of these nodes is like representing a sort of a cluster, if you like, or a group of data points. In our data, it might have been a group of patients. In this, it's a group of points in that point cloud. So we could just stop there and use the nodes. We could maybe color them in, as we did a moment ago, interpret the clusters, and sort of just point to hotspots and so on in the graph. This is done. This is sort of readily used, and you'll see this in papers using Mapper. So here, I forget exactly which paper this is from, but we are circling interesting or sort of, notable parts of this graph. However, so we're going to now deviate from this. So we, at the beginning, said that we're really interested in these topological features. We wanted to identify these features that are invariant under small transformations. So we're going to go one step further, and rather than just looking at the nodes themselves and sort of individuals belonging to each node, we want to identify topological features in our graph. So we had this example before. We don't care that it's a cup and a donut. We just care that this has a single hole. It's the hole that we're interested in, and that's the topological feature that we are looking to identify in that example. So the question then is sort of what topological features can we find in a mapper graph? And one way to think about this is you'll see that I have many different iterations of these diagrams. This is yet another one, but imagine that we have a different point cloud, data points, and we can move through the point cloud, and sort of on the right-hand side, we'll sort of build up the graph that we, as we go through the point cloud. So initially, down the bottom here, this interval here, we identify these two nodes, and these are connected. As we then move up this sort of branch here, there is a node there formed by clustering, and a second node, and that forms. And as we move up the graph, we now sort of jump from just before this hole here. We then add in these other nodes that are formed around this hole here, and we can continue. We move to the top, and we now have this other branch up here, and then other nodes formed from these points here. And I'm simplifying this slightly because, we don't need to go through all of this, and some of the details here relate to other parts of topological data analysis that we're not really covering today. But the point is, we can take that point cloud, we can identify a number of distinct features. And so we have these four types of features. So we have this downward branch shown in red here. We have a corresponding upward branch. We have this loop, also referred to as a hole. This would be the middle of our donut, or the handle on our cup. And then we have these connected components at the top and bottom. So our job with this pipeline is to identify these features, or these types of features, in mapper graphs using our data. That's what we're aiming to do. Now, the processes I've just shown you, and those sort of with the hand and the filtering and the intervals and so on, there are a number of decisions to be made, and a number of parameters that we need to decide upon along the way. So I'm going to talk through a couple of these now. So with filters, we've mentioned before, but I'm going to say more about filters because they're important. The resolution and the gain also are going to come up, and then the choice of clustering algorithm. So that's referring to within each of those intervals, we're going to decide on the clustering. Some of these can be derived automatically some of the time. For example, the resolution, if you're only using a single filter, but many can't, and we need to sort of, or rather we needed to solve this problem of how do we pick these parameters. So yes, here's yet another alternative representation of mapper. So here we have some data, in this case in just 2 dimensions here, x and y. We have this filter function. This is just using the y-axis here. We've divided this into 3 intervals shown here. and then we're going to cluster within each of the intervals. So exactly the same as what we saw before, just with a slightly different data set. And lastly, we're going to form connections between those nodes where they overlap. So I'm using this diagram because it's helpful to sort of explain where where the resolution and the gain and so on come from. So the filter we've already met, and this is a function that maps every point in our data set into a real value on a line. Here we're just using the y-axis, so we take whatever the corresponding y-axis value is of each data point. The resolution is the diameter of the interval, so it's how wide is this interval and this interval and this interval. So if you have a smaller resolution, you will have more intervals. You will get a much more detailed picture of your data. If you have a very large resolution, you will have very large intervals, you'll have fewer of them, and it'll be a much coarser picture of your data. And you might want, well, the optimal value might depend, depending on your data set and so on. Gain is the overlap between consecutive intervals. So one thing I suppose we didn't really talk much about, but these intervals will overlap slightly, and that's how we're able to form connections between these nodes. You can control that as well, and so sometimes you might want a more connected network, and sometimes you might want a sort of less connected, sparser connections between your nodes. And when I say you want, I suppose I really mean your data might require, or your application use case might require. Okay, so on to filters. So a filter can be thought of as a lens through which to view your data. So I've already said that it's this function that maps each point in your data set onto a line. The examples that I see in the literature that explain how a filter works all use 3D models. And so this slightly ridiculous picture of this kangaroo from different angles is, so the way that we would think about it is, let me jump to this rabbit. It's a sort of a different way of looking at that object. So it's a sort of a plane through that 3D shape that somehow reveals something about the structure of that topological data. And so it might be that, well, with a 3D data set, or rather a point cloud, I can just about see that you might be able to sort of think of there's a particular angle, a particular plane, a particular axis at which you want to look at this set of data points that makes sense for that object and that reveals something about that object. I struggle to make the connection to non-3D data, as we'll see in a second. So this is really, the choice of filter is really important and quite powerful. It's an opportunity to orient the clustering, orient this whole procedure around something that is relevant for your data set and your questions. So as we saw a moment ago, we are forming intervals by dividing along the filter. So whatever you set your filter to will determine those intervals. And we want to pick something that is meaningful for the data set that we're using. I don't work with 3D models of elephants, and so what if we don't have a point cloud? So I will say a little bit more about this later, but essentially we have two options. The first is what we call a data filter, and this is where you would select a variable from your data set. And this could be an outcome of interest. It could be any other variable that is somehow meaningful or significant in your clinically or substantively about your data. That it's a variable that when you order the other points in that data set along, reveals something about the structure of the underlying data set. So that's one option. And the other option is what we call a computed filter. And this is when you use the whole data set to derive some other component or dimension. So you might use principal components analysis or multidimensional scaling. You might extract the first two components and use those two components as your filters. And you can, I've been talking about filter singular mostly, but you can combine, you can have multiple filters. The the selection of your parameters, especially the filter, but all of these, it really matters. It really sort of completely determines the structure of the resulting graph. This is an example from Matteo and others' paper from 2018 showing for a given data set, it's quite small, I admit, but these are sort of quite different representations of this data, depending on the parameters that you've, depending on the input parameters. So we see that there's a sort of a loop exists in many of these, but other details come and go. And at sort of this very granular level here, the loop doesn't really appear at all. So it really matters. This might be a little bit unfair, but not particularly. The usual approach is to load your data, color it in, and then sort of vary things until the picture looks good or it looks like it reveals something of interest. We want to instead do something a bit different, and we want to optimize parameter selection for some variable of interest. So in our case, we were using an outcome of interest, and we want to identify the parameters that give us the optimal graph and the optimal clusters for that outcome of interest. Okay, so that was Mapper. Onto a couple of extensions. I have half an eye on the clock, so I will keep moving along. This is a reminder that as we now deviate even further from sort of more established Mapper, if you like, this is definitely extending beyond my sort of complete understanding. But thankfully, we have very nice and expert collaborators. Matteo particularly has helped with this. So when we earlier on looked at these topological features, I said there are 4 types of topological feature: loops, branches, up or down, and connected components. What I didn't say is actually each feature may be represented in the graph by, there might be sort of multiple cycles that lie around this feature. And what that means is we have a set of cycles, for example, a number of loops that all belong to an equivalence class, and they are sort of topologically identical. So in the diagram here, we have in the top left of this diagram, we have this hole here, and we have this green loop immediately on the hole itself. But then we have in this dashed line here, another hole or another sort of loop around that same hole. And these are sort of topologically equivalent, and we want to just pick one of these. And so when we're doing this procedure of identifying topological features on a Mapper graph, we need to, there might be sort of multiple cycles as we have here. We just want to be able to pick one of those to represent that feature. So that's one bit. And the other is that we want to select sort of robust features that are statistically significant, and we do this using a bootstrap. So this is work that Matteo and others have developed over the last few years. But essentially, we are using a bootstrap to sort of resample from the point cloud with replacement to identify features, and then only retain in our final graph those features that sort of reach a certain significance level. We use 90%. So this is sort of a new development in recent years. We don't want to, I guess 2 points here. We don't want to just try everything and see what works, and we don't want to end up with and interpret features that are sort of might appear just because of small variations in our data. And lastly, on this sort of set of extensions, is we want to be able to use mixed data types. So far I've been talking a lot about point clouds and 3D shapes, and what we can also use as an input is a distance matrix. So if you don't have a 3D shape, but you do have a, as we did, we have a sort of more standard data set of rows and columns. We want to construct a distance matrix, and then we can input that into the Mapper algorithm. And so to do this, we're using the Gower distance matrix, which is essentially going to use an appropriate distance measure depending on the type of variable. And in the interest of time, I'll say no more. But this is nice and flexible, and it allows us to incorporate mixed data types. Okay, so lots of talking, onto the actual pipeline. So just to recap, we're using the mapper algorithm. We're interested in identifying statistically significant representative topological features. And we're interested in sort of just selecting within those features clusters of interest. So I have this diagram, this photo here. This is in the Cairngorms in Scotland. It says, take care, you're entering remote, sparsely populated, potentially dangerous mountain country. And I feel like this is what we were doing in sort of starting to come up with a pipeline. And I feel like, as you will see, and I'll maybe come to the end and maybe in the discussion, it was often hard to know which direction to go. Like there is no textbook yet that is sort of applied topological data analysis for sort of biomedical research. And so a lot of this was sort of thinking through what are our options here, what are the implications of taking this route versus another. This is, this was a process, and you're seeing the sort of the end result. Okay, so this is the pipeline in a nutshell. So we're going to construct a distance matrix. I won't say much more about that. We then are going to enumerate all combination of the input parameters. I'll talk about that. We're then going to use mapper to identify these topological features, and that's what we've just been talking about. And then these last two steps I haven't covered yet, and so I'll say a bit more about those. As I said, we're going to optimize the sort of selection of parameters, the choice of filter, the gain, the resolution, and the clustering algorithm for a chosen variable of interest, like an outcome. So this process here is essentially identifying what are the optimal features for our data set. And lastly, we want to be able to visualize this and summarize this effectively. So we have a number of different input parameters, and for each of these we have several options. So we considered a number of filters, both data filters, so baseline values of the Madras and BDI scales, some computed filters based on PCA and MDS, and some combinations of these. We considered 4 values of gain, representing different levels of overlap between the intervals in the mapper graph, different values of resolution, and then 2 different clustering algorithms to consider within each of those intervals. So this adds up to quite a large number of different input parameter sets that we need to consider. We then, for each set of parameters, we're going to compute the mapper graph. We're going to identify the representative topological features. And of those, we're going to select just those that reach a certain significance level using the bootstrap. So we're sort of removing non-significant features. We're then left with many, depending on the number of parameters options you've considered, candidate mapper graphs. At this point, we then removed any graphs that have no significant features. And this point here is important. We extract representative features from each graph. So I just want to emphasize this point here. So what we've done so far is we have all these different parameters. We've run mapper many times for each set of parameters, and we've resulted with like 3,000 mapper graphs. And after removing the ones that don't have any features, we are then going to identify and pull out the topological features from this graph. So our sort of unit was the mapper graph, and in this step, our unit becomes the features themselves. And from this point on, when we talk about clusters of patients or clusters of data points, what we are referring to are the features. So that was a point worth emphasizing. So now instead of 3,000 graphs, we have probably even more thousand features. Each of these is a particular cluster of patients or individuals or data points. We then removed features containing everyone or no one, or sort of more than 95% or less than 5%. Okay, so at this point, we have a long list of candidate clusters, and we want to select the optimal cluster. And to do this, we are using homogeneity. So homogeneity for each feature with respect to this chosen outcome of interest, as well as percentage improvement in homogeneity compared to the overall sample. So what do we mean by homogeneity? So for binary outcomes, we're using Gini impurity. So it's given here, but essentially this is going to range from 0 to 5. Lower values would be better. In our case, we want to identify a cluster that is sort of distinct compared to the rest of the sample. And so we're looking to sort of find the lowest value of Gini impurity from across those many clusters and those many features. For continuous outcomes, we're using the standard deviation. So this last part, so then what we've done, we've sort of run Mapper many times, we've extracted the features, we've sorted them based on homogeneity, and we've selected the top 5 or so. Why 5? I don't know. I think we might actually have 20 in the final analysis, but we select some number of the sort of optimal features to consider, and we then want to visualize them and summarize them. The visualizing I'll show in a second. In terms of summarizing, we consider a number of things describing differences between the input variables for members and non-members on each cluster, statistical tests of these differences, but also using XGBoost to predict membership to that feature, that cluster, based on the input variables. And we were going to report, or we do report, the feature importance for each input variable from your dataset. In terms of visualization, this went through a number of sort of iterations. This was an early example showing a graph with a number of features, and we sort of label the features and color them in, L here referring to loop, and there are a number of different loops. At one point we had, I don't think we're still using this, Raquel, but at one point we had a Shiny application to essentially do this as well, and this sort of allowed you to experiment with different thresholds and vary a few other things. We didn't use that really either, and what we ended up with was sort of a lot simpler, and this is just really draw the mapper graph and highlight the significant representative features on that graph. This is, I think, a lot of topological data analysis is visual, and so we started out with lots of attempts at how can we use these pictures, these visualizations to improve the interpretation. Ultimately, I don't know how useful these pictures are, but we can come back to that in discussion. What we ended up with, you'll be very sad to hear, is Excel files. So for each of, when we run this pipeline, what it outputs is an Excel file that summarizes in a number of different sheets along the bottom here, the optimal clusters, that is the optimal topological features for your input data set. And there are a number of summaries and the graphs in there as well, and so on. But that's what you get out, and really we should probably do something better, but this is a compromise of time. Briefly, because I'm aware of the time, haven't really talked about software specifically yet. The sort of big name, if you like, in topological data analysis, especially for sort of business applications, is a word I've never said out loud, but ASD. So this was co-founded by Gunnar Carlson in 2008, the person who wrote Mapper originally. And they used to have academic licenses, I'm told, but right now they do not seem that oriented to, well, I think their applications are more sort of business oriented rather than what we're trying to do. So everything that we're using here is from open source packages. The big ones are Goody, that is, I think, really the backbone of most open source topological data analysis. So that's in C++ with a Python interface. Then there are two packages written by Matteo Carriere, who is a co-author on the paper and part of the working group and has been invaluable throughout this process. So sklearn, TDA, and SnapMapper. Some of those packages have been superseded by Goody now, but importantly, they include things like the identification of representative features and the bootstrap. So we use both of those. And then as well as sort of standard Python libraries like pandas, scikit-learn, and so on. I'll leave this here. I won't say anything more, but there are a few other packages available in R, in Python, if you're interested in this. Computing, so if you have about 3,000 mapper graphs, and for each of those you need to run a bootstrap, it gets really slow. Thankfully, this is sort of readily parallelizable, and Rosalind was invaluable for this. Here, I'll circulate these slides afterwards, but this is just showing for a single multi-core PC, we can use GNU Parallel to parallelize it. On a cluster like Rosalind, we can do something similar. And yeah, there are some challenges there. I can maybe talk about that later. Okay, so some conclusions and some strengths. So the big one, I think, is this ability to choose a filter to orient the clustering. And so this is, I think, powerful and novel and really quite useful. And it allows you to sort of have a say, have sort of some variable, a number of variables to orient the process. The use of the bootstrap, as we've seen, to exclude non-significant features. We can use mixed data types with the Gauss distance matrix. The procedure of the sort of using a grid search to select the optimal clusters and the optimal features based on homogeneity in our case, and this avoids the sort of trial and error. Let's just vary all the parameters until we see a picture that we like. Visualization, we did that. I'm not sure how useful it was. And then lastly, this use of things like XGBoost and other statistical summaries to really interpret the clusters statistically rather than just sort of visually and coloring. In terms of limitations and challenges, the specification of the parameters to explore, or the grid of parameters to explore, is somewhat arbitrary. This could be improved with someone who knew more about topology, I think. We did obviously work with our collaborators on this, but I think at times you might not always have a good idea about, okay, what values of gain do we need, or what values of resolution do we need? And you are sort of left with, we'll try a few and see which of these works best. The parameter tuning can be slow, as I've said, and relies on parallel infrastructure, and we haven't tried this yet on very high dimensional, very large data sets. We're not quite sure how this will work. The code isn't packaged very well, I would add. So it works for the data set that we've used it for, but it would need sort of adapting to work with any input data set. It's complicated to explain. I think it's a real thing in that we've had a whole talk just talking about the pipeline with no results. We have a paper about the pipeline and no results. Why not just choose a simpler method that doesn't raise so many questions from reviewers and so on? And we haven't talked much about performance, but preliminary work that we've done so far and also reviews elsewhere suggest that this is sort of better or as good as other things like k-means and MDS and so on. But there is some work to be done there. Last slide from me. Lessons, don't be first. So I think this is related to that picture of the me hiking in Scotland. And often I felt like we're sort of asking, you don't want to be the first person sort of trying to navigate, maybe you do, but it's quite challenging. And I think I'm still, as I said here, I'm still waiting for the Applied Researcher's Guide to TDA. I would love that textbook to come out. And then keeping up with the literature is really hard. We started this, as Raquel said, a couple of years ago. And in that time, things have been moving so quickly that sort of in the time that we figured out this pipeline, I'm pretty sure things have moved along again. In terms of next steps, we are in the process of applying this pipeline to the GenDep dataset. So actually using it and hopefully talking less about the methods and more about the clinical implications of those clusters. And secondly, as I said at the beginning, using TDA for prediction. And so how can we use topological data analysis with repeated measures to improve prediction? But I will stop talking. We have a little bit of time for questions. Thank you to collaborators, particularly. Thank everyone for listening and happy to take any questions that I can answer.
Thanks very much, Ewan, for this excellent talk. So yes, as Ewan said, if there is anyone that would like to make any question, please go ahead. Yes, Derek?
Hi, Ewan. Hi, you can hear me now?
Yes.
Yeah, no, thank you very much. Very interesting talk. I like this TDA. I mean, it's quite close to some of the things I worked on in terms of network analysis and object data analysis. And I feel that some of your concerns there are things I have encountered before. At some point, you say, how useful are these graphical visualizations? I think this is something I've felt sometimes. We're transforming things into graphs, and I felt that, you know, it was, but I guess when it comes to new forms of analysis like this, there are really two different ways to go about it. I mean, either this is something which is outperforming existing techniques, and I think with clustering, it's going to be hard, I think, to produce, because clustering is massively has been somehow, you know, from the 70s. This is a the different techniques for clustering, and with network analysis, we were doing a similar thing. We were doing community detection, and I'm not too sure exactly how much this has outperformed existing very basic clustering techniques like k-means, for instance. K-means, in fact, has incredibly good properties. But so then the second angle is for the new type of data analysis to answer new questions or to somehow shed new light on this. And I felt that certainly this is the interesting thing about TDA. But so when you're representing those geometric objects, though, it feels that traditionally topology has been about identifying holes. So I think there is something called the Gini numbers. At some point you talk about Gini, but I think Gini numbers is more or less counting the number of holes in a particular geometric object. So, but what else? No, maybe I'm wrong. Raquel is saying no, but it's sort of.
Can it be Betty numbers and not Gini
Betty?
numbers?
You're right. Betty, maybe Betty. You're right. It's a Betty number. Betty Gini. That was easy to confuse. Okay, but what? But I think because you've mentioned the word topological features a fair number of times. But besides clustering, beside the holes, I mean, what else is there?
Thank you. So these are good questions, and Raquel can jump in when as soon as they go wrong, which might be quite quickly. But from my understanding, there isn't anything else. As in, we are looking for those features, and the biggest challenge I have with this work is translating from the sort of, if not the theory, but certainly the applications of this work in the topological literature, which is all about shapes and Betty numbers and other things, and translating that into, okay, but I just have a grid of rows and variables, and what does a feature mean in the like what is a donut in the context of a real, well, not real, but in the data that I'm going to be using. And I don't have a good answer for like, I mean, the short answer is the same features. It's those 4 features. That's it. But in terms of like in my head, at many points in this process, there is I am continually challenged to connect up what we are doing in theory, or what we're doing, essentially what the code is doing and what these packages are doing, with like the real world interpretation of that. And there's for me, and we'll stop talking in a second, but like the biggest departure is always about this point cloud, and the fact that you have a point cloud and we have 3D shapes and things, and then I understand Mapper in that context, but we often or almost never, in my experience, are using point clouds. We have data sets, and so then it's sort of, there's always a leap in sort of conceptual, just like, trust me, we're going to definitely find these topological features, the loops and the connector components and so on in this data set. But there's a sort of gap there in the understanding. The point about, and I think I'm just also seeing Jodie's question in the chat about how does this compare. I feel like we should do more on this because we, in our application so far, we've compared it to k-means. So in the output and in those Excel graphs, documents, we are comparing the homogeneity achieved with k-means on that same data set with the mapper graph, and what we're finding is sort of performs as well as k-means, which is not really a lot to shout about. And I'm not, as you say, I agree with you that given the interest in clustering generally, it's unlikely probably to, I mean, Raquel, you can step in, but I agree that sort of beating other clustering algorithms on performance might not be the aim, but I think there are other advantages of Mapper, especially this like ability to incorporate a filter that is worthwhile. So I don't know, Raquel, I don't know if you wanted to say anything.
No,
performance.
I'm talking by heart, but I think that the top cluster in outcome homogeneity was found using Mapper actually, and that k-means, the cluster that we got using k-means, were actually throwing lower values on homogeneity in remission for patients. But it's true that we haven't done more work on simulation, that this is what we should do really. But others have done that. And looking at the literature, we found that other works comparing Mapper to hierarchical clustering, also to k-means, BB-scan, or other clustering methods like single linkage and complete and average linkage clustering, really found that, and when data was basically different, so all the different methods were producing similar clusters. But when data had small variations, Mapper was found to be more sensitive. And I think this is because of the step of resolution. So if you remember the slide from Ewan's talk, when he was showing the hand, there is a point when he said, we split the data by beans, and this step is key, because depending on the beans that you consider, you are putting a higher resolution on your data. And so you build a kind of continuous division of your data, and then you look at it with finer detail. And I think that this is quite different from the rest of cluster methods that starts with all the data as a whole, and they try to do a cluster of the big thing. And in this case, it's like that you are putting a lens on your data, and you allow your method to see the data with more detail and to get these topological features from there. And the other thing is that you are not looking at geometry, but only to topological features, which is quite specific. It's not just only holes, it's connected components that sometimes are not detected by standard methods, especially by k-means, and holes and cavities, because remember that we are going to very high dimensions. We don't even know how a hole is in dimension 1000. And so I think that here, there's a lot for TDA to find out as well. Yes, maybe we're positive, but I think the first results that we got are throwing some positiveness for us to believe that this can show, or this can show a bit of improvement over the rest of methods. Okay, so
I think Nick had a question, but yeah, no, I mean, yeah, emphasizing, I think the holes, I mean, because this is something that nothing else is doing. I think that certainly is definitely, is not just promising, is obviously is looking at the data from a different perspective. So it's obviously an improvement. Yeah.
Nick,
but I mean, with looking at the data from a different perspective, what's the dangers of overfitting? Like if you're trying to search for something, this shape in the data, and you're saying this shape does exist, how then specific is this shape to the data you're using? And especially, I guess, if you're using the small data set with a lot of hyperparameters, I'm imagining for TDA, you need really large data sets to get something that's more generalizable.
Yeah, I agree. And I think, yeah, that's something that we would need to, well, in this application, and I think the intended output of this pipeline is exploratory. We have a data set and we want to, we are interested in identifying potentially relevant clusters that we would then go and do more work on. But I agree, the concern remains, yes, you need large amounts of data. And I guess there's just that unanswered question of where does this fit in terms of other methods. So we are using TDA for prediction, and the sort of approach there is quite different. The grid is well, we're not really doing grid searches in the same way there. So this particular application and this quite wide, let's consider all the possibilities, all the features, all the graphs, is only, I think, the intended use, or is only good for sort of exploring the data and trying to uncover things that you would then interpret in a sort of, well, yeah. So
yeah. I think that's it. It just seems so different. It doesn't seem like comparing with like conventional clustering methods is doing yourself justice. I think it's all about the right research questions. It's really interesting, and it's good that there's this different way to look at the data. But I think we need to sort of discuss what can we get from this? What's it giving? It's obviously giving us something that other techniques can't. But fitting that all into a pipeline. The next step is a great pipeline and great work you've done. But this next step fascinates me. So I'm looking forward to chatting, I guess, with you and Raquel offline about that at another time.
Yeah, definitely.
Okay, thank you, Nick. Is there anyone else wanted to ask to Ewan, comment something? Okay. So Daniel, please, yeah.
Just a question. I mean, what about missing data? How do you handle? Did you think about handling missing data? Because that's the biggest problem in psychiatry. Was it a question at this time?
I forgot to put something in the slides. Yeah, that was it. So we did, but not very well. So we're imputing data at the outset, but I think it's also like related to the previous question and sort of how do you sort of use these results and interpret results. So at the minute, short answer, we are imputing missing values. I'm trying to think the exact point we do it, but once, short, and we're not doing it sort of repeated or anything like that. So I think that the sort of that question of what do you then do with these results, and Raquel, I think is something that we can sort of think about and work on. Most applications of TDA that I've seen are all about prediction modeling, and sort of I've seen less that, well, the ones that use it for clustering like this are more of the sort of color it in and interpret the pictures. And so I think we have a question to answer is like, what do these results mean? What can we say about the resulting, the final chosen optimal clusters? But the short answer is we impute once. We did have some missing data.
It might be useful to look at imputing your data in your bootstrapping process. So if you bootstrap and then you impute each time, so you can see how stable your results are. I think Jonathan Bartlett wrote a paper about it with cross-validation. So you might have a look at it, but I think it's a topic which still needs a lot of work in prediction modeling. And I think the topological data analysis looks really interesting. I think especially to identify new features. And I think especially in high-dimensional data like variable data or something, it would be really interesting to see what you can do with it and how to interpret the features.
Yeah.
Thanks, Daniel. There's a comment in, there's first a comment from Nick in the chat. He's asking if TDA could be used to impute. So I think that with Ewan, we haven't done anything similar to that, but we are developing some work on imputation with our PhD student, Yi Yang, who's also attending. And what we have found for the moment is that TDA is being used to compare imputed versus non-imputed data. So it's like a method to assess the quality of imputation of other imputation methods that are not related to TDA, right? So what people are doing basically is to check the topological structure. And so if in the complete data set, the non-imputed, you find that you have this connected component or this whole, so you would expect that in your imputed data, the topological structure should be preserved. So this is a way to use a TDA, at least to assess quality of imputations. But I'm also interested in understanding and trying to develop methods and think of methods of using TDA to input data. So this is something we could think about, actually. There's another question, Ewan, from Xiao Xiong. So he said he thanks you for the talk. If I understand right, novel nonlinear features will be generated using TDA. Do you think similar feature can also be learned through models such as autoencoders?
Yeah, so my answer is yes, and I don't know. So maybe we can talk afterwards. But yeah, I'm not familiar enough with autoencoders to
Okay, thank you. And then there's a question by Richard Ensley. It's interesting work. So we should have a session on whether missing data is the biggest problem in psychiatry. You're right, Richard. Okay, is there anyone else who would like to make a question? If not, we will leave it for today. So it's now 11 past 5. Excellent. So let's finish the talk for today here. Thanks very much. That was super interesting, a brilliant talk. Thanks everyone for joining and for your discussion and questions. And yeah, we'll see you next month again, the 4th Wednesday of the month here in the prediction modeling seminars. So thanks everyone, and keep safe and see you soon. Bye. Thank you.